test cross, okay, Adam Suyang, you do the test cross, right? You are crossing the homozygous recessive with the which is this one is a heterozygous dominance alleles. So you can get about the different phenotypes results, which is 50% of them will be purple flower, 50% of them will be white flower. But if you do the cross, if you cross them with the homozygous dominance, if you cross this homozygous dominance with the homozygous recessive, you will get all of them is purple flower. Because you produce about, the genotype is one big B, one small b. Okay, so this is test cross. So by looking at the offspring phenotype, right, you can trace back, you can know. So actually your parental phenotype, which is the dominant one, is either is homozygous recessive or heterozygous recessive. It's either homozygous dominance or is a heterozygous dominance. You just want to know this, so that's why you do this test cross. Okay, which is this one. Dominant phenotype, but it's unknown. You are not sure it's heterozygous dominance or is homozygous dominant. So that's why you need to do the test cross with the homozygous recessive genotype. So this is test cross, okay? Because they are actually crossing this heterozygous dominance with the homozygous recessive alleles. Okay, and then the second question, you talk about what? You talk about the how they do the selective breedings, right? When they do the selective breeding, they are doing what? They are doing this one, okay? They are actually select the parent plants. So the parent plants, right, they will carry the desired features. They will carry these desired features. So for example, that the offsprings, they will just grow faster, or the color is more attractive. They just select the features. So, and then they will continue the process of crossings and selecting for many generations to produce a pure breeding varieties. So the keyword is you are actually selecting the flower which carry the desired features. And then you are selecting the offsprings just showing these features, and then you continue to cross them, select them. So that's why I just directly paste the answer for you, because you can read. I want you to read, and then know what keywords to write in the exam. Okay, yeah, we continue to do about the genetics, because you should know how to draw the genetic diagram. Because this chapter 70, you are not required to memorize too much thing. You just need to know how to, you just need to know how to write the genetic diagrams and also some of the definition on it. Okay, yeah, we'll part of this one, right? There'll be two alleles, okay? There'll be two alleles. So you can see, right, the first one alleles will be this one, the red flowers, and then the second alleles will be the white flowers. Okay, one is red flower, one is white flower. So they're asking you to say the genotype for the plant that's heterozygous for these genes. So we have red flowers and the white flower. So you can see, one A and one N. Okay, yeah. So that's why you can heterozygous. So heterozygous refer to what? Heterozygous refer to both of them are different alleles. One dominance, one recessive. This we call heterozygous, different form of allele. So describe the phenotype for the heterozygous carnation plant for these genes. So how is the phenotype if you have about one A and one N? So which is when the red color mixed with the white color, you get about pink color, right? So you can see this pink color because both of them are, they are actually showing the color. So actually there's another term because what we call it as a co-dominance. So you can see, right, this one, co-dominance. So at this point, we're looking about this term co-dominance. Yeah, it's called as co-dominance. This co-dominance, it just refer like this. Let me show you co-dominance, right? Which is both of the alleles, they will express themselves. So like this. You see, you have the red flower, which is both of them are just in the red. Okay, the big R. You have the white color flower, which is they have a small r, and then pink color. You can see pink color is between the red color and the white color. So this is their genotype. One big R, one small r. So this is codominance. Then example of codominance is not included in the color of flower, but also include in the blood type. You know, our blood type also example of the codominance. Because all of the alleles, right, they can express themselves. They are not being suppressed by the dominant alleles. And then so that's why you still need to draw by a geometry diagram. So just like, okay, how you able to draw? Very simple, just copy down. Copy down the gametes, okay? Okay, just copy down the gametes. Like this, okay? And then you have this one. And then now you need to do Punnett square. Okay, the Punnett square, right, actually it just like this, okay? You can or you can draw like this. And then this will be this and this. So at the end, right, you get about the combination, which is this one. F A, yeah. And then F A again. And then you have a F and also F and again. So this is your genotype. Okay, so this is your genotype. So in the genotype, right, you can draw something like this. Yeah, you should write about the answer is just like this, okay? Just write down this at here, at this column. And then how is the phenotype? The phenotype, right, you need to describe, okay, you need to describe about the color, okay, the color. So the gamete is like this, okay? And then the offspring genotype, okay, you should summarize. Yeah, you should summarize. So you will have this one. Just write 2 enough for you already. Then another one will be this one, FA, FN. So half of them will be is about 50 to 50%. So how about the offsprings phenotype? The FA and FA, right, is actually have what? Have the red flower. Okay. So it's about red flower and the pink color. So how's the proportions of the pure breeding carnation plants? Pure breeds means what? Pure breeds means it's red color or it's white color. Okay, that's pure breed. Pure breed refer to the both alleles, right? They are the same, which is both have A and A. So it's about 50%. Okay, so you just write 50%. Because pink color is not pure breed. Pink color, right? They always have a mixtures, okay? Mixtures of this dominant allele and another dominant allele. So it's not pure breed already. And then, okay, for this number 3, right? Yeah. Number 3, okay, this is a Punnett square. You can see, you just complete the diagram below to show how the sex is determined in the cats. So you have the male cats with the female cats. So how is the gametes in the female cats and the male cats? Okay, this is, okay, now this is different because this is, we talk about what? We are talking about the sex-linked gene. Okay, sex-linked gene. So what's mean by sex-linked gene? Sex-linked genes is referred to something like, because right, in your 6 chromosomes, you have about X and Y, right? X and the Y. So this is your 6 chromosome. So at the start, you are boy, so that's why you have the 1 X and 1 Y chromosome. And then I'm girls, so I have 2 of them is XX. So this is in the mu, okay, mu. 1X, 1Y. And then this is in the female. Both of them are contain XX. So what will happen for the male cats? Okay, they have a 1X, 1Y. So at the end, you have about XX, XY, okay? And then this also XX, XY. So the offspring ratio will be about 50% of them is male. And then you can say 50% is female. Yeah. And then for this one, okay, the genes for the coat color is located on the X chromosome. So you can see, right, whenever you see any of the genes, if the gene is located on the X chromosomes, this refer to what? This refer to these chromosomes, they have what? They have a sex-linked gene. This sex-linked gene is different from the autosomal gene that you learned just now. Okay, because this gene, right, they are specifically located as the X chromosome. They're just located at the X chromosome. So you have this B and the small b. They just at the X chromosome. So that's why when you write the sex-linked gene, right, you should write in this way, like this and like this. Like this and like this, okay? So this is a bit different. Sex-linked gene. And then one thing is, when you talk about this sex-linked gene, right? Usually is they will only affected in the male. Female is not really affected. Okay, later I'll show you. Okay, but and then you should start to write the genotype. Okay, 1, 4, and 5. So you know about 4 and 5, right? Okay, 4 and 5 is affected because it's affected, right? So affected is about the gene is okay. Is black in color. Okay, it's black in color. This one, black in color. Black in color will be the big B. Okay, so you can see, right, the male color and the female color. Male is about XY, right? Okay, XY. Okay, where's my pen? Oh, my pen is not functioning. Okay, it's okay. Later I'll show you. So you can see, right, this 4 is the male. So male is about XY or XY. So since it is in the black male is about what? B. So it will be the X, capital B, like this. So this is number 4, okay? We can see X, capital B, and then Y. Then how about in the pet number 5? Pet number 5 is the female. So that's why both female, right, they will have a B, okay? They will have this B on the X like this. So you should draw like this, okay? Both of the X chromosome, right, they will have the B, the genotype B. So how about the cat one? Okay, cat ones, they are orange male. Orange male, right, they will have small b. So since it's orange male, it's orange male, right? So it's XY. So the B is small b, because small b is orange color. Yeah, small b is orange color. Big B is black color. So you just write like in this way. Okay. And then this is a DNA. Yeah, DNA. You should this is very simple for you because you just need to list out the complementary base pairing. Yeah. Okay, some of the question I think we didn't discuss yet because you haven't learned it. Okay, today I just want to show you about how you're able to draw the genetic diagram in the correct sequence. Okay, so for example, another question they were asking you for this chapter is about, they will ask you about what? They will ask you about the protein synthesis, right? The way on how the body cell able to produce the proteins. So yeah, so those are the process. So they give you about how many marks? 3 marks. 3 marks, right? So you should say that like inside the nucleus, they contain the DNAs. So you can see, and then the DNAs, they have the base sequence of the genes found inside the nucleus. So what they do is they will produce a messenger RNA. So the mRNA, they just carry the genetic instructions from the DNA, and then they will go to the cytoplasms, and then they will attach to the ribosomes. So that's why the base sequence on the mRNA, okay, they will, they will detect the sequence for the amino acids. Yeah. And then later, they will produce the polypeptide change. So polypeptide change, they can fold and they become proteins. Okay, so actually this process, right, is also written in this answer. You can refer to here. See? Okay, describe how proteins are made in the cell. Either they will give you 3 marks or 4 marks. So you can say that, I think this is better, okay? This answer is better. The mRNA just copy from the gene. You should write about this one. Transcriptions, you should write this term, transcriptions. So the genes coding for the proteins, right, they will just remain in the nucleus. They just remain in the nucleus, right? And then the mRNA, and then the mRNA molecule made in the nucleus, and then they will move to the cytoplasms. And then the mRNA, right, they will pass through the ribosomes. The ribosome, they will just translate. So you can see, right, ribosomes undergo translations. So when they undergo translations, they will join the amino acid together, become a polypeptide change. So this is how they will form the proteins at the end. So those are the keywords. The mRNA, the transcriptions, go to the nucleus, bind to the ribosomes. So ribosomes, they will join the amino acid together, form the protein. So those are the keywords. Okay, let me show you another questions. Yeah, and then how we able to describe the structure of DNA is the DNA molecule is double helix. If it's two-stranded, right? Just two-stranded join together, so they call it double helix. And then you should describe the base pairing, okay? Like A to T, C to G. And then those base pairing, right, is also joined together by the hydrogen bond. So you can see, right, there will be the requires what? Complementary base pairing, okay? Like A to T, C to G. So actually there will be the bond holding them. I hope you know this. So this bond they are known as what? They are known as the hydrogen bond, okay? Hydrogen bond. Yeah, hydrogen bonds. Okay. Another kind of question I want to show to you is, let me show you about this, some of the questions. Sorry, this chapter 70 is a bit too much, okay? But yeah, in the actual exam, they will just come out in this kind of questions. Just similar question only. I think I should show you about this. Okay, it's okay. Okay, let me show you about this one. So just now we learn about test cross, we learn about how to write. We learn about how to write the genotype, okay, the genotype for the answer. And then, yeah, for this one, okay, I want to show you about a thing. Where is it? I can't find it. Oh, it's at here. Sorry, it's at here. Okay, this one. Okay, this one, this note you can have a read. Okay, you just have a read. Yeah, so just remember that the DNAs combined with the histone proteins become the chromatins. Okay, the longer and thinner strand we call as chromatins. So chromatins, they can further coil together, become the chromosomes. So the chromosome will become like this, okay? So each one chromosome, right, they will have 2 sister chromatids. Each one chromosomes, they will have about 2 sister chromatids. Okay, and then this also, yeah, I think we discussed about this before already. Then the transition and translations, and then let me show you about this one. Okay, so that's why when you write the genotype, right, one genotype, okay, it must be contain 2 alleles. Either is too big T or too small t. If it is a homozygous dominance, right, it is called as the too big B. 2 of them are big capital letter. But if it is a homozygous recessive, both of the capital letter will be smaller, okay, small letter. or it is a heterozygous, you'll be like this, one big letter, one small letter. So you know that every alleles, right, they will have the same gene functions, but they just express themselves in different way, different physical form. And then I want to show you about this one. Yeah, this one, okay. The genetic questions. So at the student, how you can answer this genetic question? Okay, whenever you answer this question, right, first you need to identify whether this question is asking about autosomal disease or it is asking about the sex-linked disease. You know the autosomal disease and sex-linked disease is different. You need to identify this first. It's because that if they are asking about autosomal disease but you just write it as sex-linked, right, the answer will be totally wrong. You will not get any marks. Okay, so that's why you can read on the questions and then you should identify, because for the autosomal disease, right, for this autosomal disease, usually male and female, right, they are equally being affected. They have a same, we call it the same probability of getting affected because it's autosomal, it's not sex-linked. But if in the sex-linked gene, right, in a sex-linked disorder, okay, or sex-linked gene disease, usually the genes, okay, the defected genes will be located on the X chromosomes. Why? It's because that the X chromosome, right, they are larger in their size. Do you know? And that's why you have about X and Y chromosome in your body. The X chromosome, right, is much more larger compared to the Y chromosome, just like this. It's super big, right? Maybe the Y chromosome is just like this. Okay, it's just smaller. So this is X, this is Y. So there will be a lot of the defective genes, because of sex-linked genes, they are actually located at the X chromosomes, not at the Y chromosome. So that's why, because female they carry both X chromosome, right? So that's why they will usually a carrier, the carrier for the sex-linked gene. And then the male, right, because they only have about X chromosomes. So if the X chromosome are unlucky, they carry the defective genes, they will directly show the symptom, because they only have one X chromosomes. But in the female carrier, okay, because they have about two X chromosomes, so for example like this, okay. So for example, female mostly of the female will be the carrier. You know what's carrier? Carrier is, for example, like you have about carrier. Okay, for example, right, the sickle cell anemia, okay, we talk about sickle cell anemia. Okay, not talk about sickle cell anemia. Okay, color blindness. Let me show you. Okay, color blindness. For example, the A. Okay, A is a color blindness gene. You know, color blindness, 色盲, color blindness. You know, color blindness is a sex-linked gene. It's a sex-linked gene because this gene, right, is located at a sex chromosome, and then most of the male will be affected. So color blindness, okay. So big A is a color blindness. Then small a will be the normal patient like you and me, okay, it's normal. So in the female, because you have two copy of the X chromosome, so if one of the X chromosome, right, they carry that A, which is the big A, carry the big A, right, and the small A. So this is a carrier. Okay, this is carrier. So carrier will show any symptom or not? No. Carrier will carry that defective genes, which is this one, but they will not show any symptom. But what the carrier to do is when they marry with another guy, right, they can pass this defective genes to the next generation. So if they have a son, and then the son are so unlucky, they will inherit the gene. So they will have the color blindness. So that's why female usually is a carrier. Female will only be affected if both of the X chromosome, right, they'll be like in this way. If both of the X chromosomes, they will be carry this color blindness gene, so that's why they will only get affected. Okay, so usually it's male affected because they only have one X chromosomes, like this, okay, X, Y. So if you are healthy, right, you get like this, okay? You are healthy, you are normal. But if the genes on the X chromosome, you get this one, you become affected. You show the symptom, which is a color blindness. So that's why the sex-linked disease usually affected in the male only. So male is being affected because they only have one X chromosomes. If they get the genes, right, they will just show the symptom. But in the female, right, is carrier, and then they have about two X chromosomes. So another X chromosome, if they carry the normal gene, right, they will help to suppress. They can suppress the effect of the defective genes, so they will not show symptoms, but they can transmit the gene to the next generations. So that's why, as well, in future, if you want to get married, okay, I will advise you to do the genetic testing. So you can trace back your family, like maybe your family get what kind of the genetic disease, so you can prevent anything happen. And then you should study the question, okay? Maybe they ask about this one, okay? The disease itself, right, is inherited. Oh, okay, the time is already passed. Okay, I just need some minutes, okay? Just give me 5 minutes, a little bit. So you should look about either the disease is inherited by either its dominance or the recessive alleles. Dominant alleles or recessive alleles. Okay, because you cannot think all dominant alleles is a good one, okay? Because sometimes the dominant alleles can be the alleles which carry the defective gene. So that's why you should read on the title. Okay, so which disease is caused by the recessive or the dominance? So for example, you can see like in this question, okay? Hemophilia, okay? Hemophilia is a sex-linked blood disorder. So they show you about sex-linked, okay? Come on, sex-linked. So, and then they show about only the male affected. Because the male only carry the X. So that's why only the male is affected. So what you can see is, okay, say the genotype of the people identified as PQR. So the P is what? The P is the female, right? It's a female. So the female, they can be the normal or they can be the carrier. So because of their offspring for the male, right, they will have the affected one. So you can assume that maybe the female, they will become the carrier. So the carrier is, they will have one big H, one small H. Okay, one big H, one small H. Then how about in the Q? Okay, Q, because it is the male, male. So and then it affected. So that's why it's X, Y. Then the X must be the small H, because it's contain the hemophilia. So you can see the Q is small H. Oh, sorry. Okay, the small H. And then how about the R? R is the normal one. Okay, the normal patients. So R is the XY. The XY, they just carry the normal gene, which is the big H. Big H is normal one. So during the exam, right, you can, when they ask you to draw about genetic diagram, you can give a symbol. Okay, you should write about your symbol is referred to, for example, like this. I write that the big H, right, is referred to normal. The small H, right, is referred to hemophilia. When you write this, the marker, when they mark of your answer, they can understand. Because they already say that the normal others is represented by the big H. The mutant one is represented by the small H. So that's why you need to follow the questions. Okay, so that's why you need to do the crossing, okay, the crossing. So you have about female and the male, right? The female is, it's carrier, so you have a small, one big H, one small H. Male is the normal one, normal one. So that's why you have about, at the end, how is your genotypes? You get about like this, okay? X, H, okay? And then X, H. And then another one is X, H, and Y, okay? And Y. And then X, H, X, H. The other one is this one, X. Oh, what happened? X, Y. Which is they have H, like this. So that's why you have a normal, a female carrier. or the affected male. So this will be the affected male, right? So this male will getting that disease. So it's about 25% because they're asking about the probability. So at the whenever they ask about probability, right, they are actually asking about the percentage. You can write about 25% or you can write 0.25. Yeah, it's up to you. Okay, so what I mean by sexing gene is refer to the genes, the defective genes only located at the X chromosomes. Yeah, so that's why the male, right, is more affected compared to the female because the male only have that one X chromosomes. So if they get the defective genes, right, they will just directly show the symptom. Okay, so I think that's the class for today. We can continue the next class. Okay, the homework is the objective question. Okay, I have sent in the group. Yeah, this one. You can try. Okay, chapter 70 objective questions. Okay, happy study. Bye. Good night. Bye.